Welcome to the first annual Symposium on Mitochondrial Health, Healthspan, and Aging. Mitochondria and the Emerging Science of Salugenesis by Bob Naviaux.
It's really my pleasure to be here today, and I want to thank Sandeep and the whole Blue Oak Nutraceuticals group for really putting this symposium together. So I'm going to talk to you about mitochondria and their role in healing. Okay. And so a secondary title for this is Salugenesis and the Cell Danger Response, how signals in the blood can change mitochondrial function and lead to health or symptoms of chronic illness. So I have a few disclosures. I'm an unpaid scientific advisor for the OMF and autism community, ARI, and a few other places. And what I want to really talk about is that my lab really only studies one thing. We study mitochondria from conception to old age. But we find that it's connected to all life on the planet. There are three vocabulary words that I'd like to start off the talk with. One is salugenesis, and the definition of that is a multi-step process of healing that's triggered by the cell danger response, coordinated by mitochondria, and maintained by extracellular ATP signaling. The cell danger response is the system's biology response to environmental threat that's coordinated by changes in mitochondria. So the CDR is a whole systems response that starts with mitochondria in the cell. Purinergic is a name for the signals that are transduced by a family of cellular receptors that are activated by extracellular ATP, UTP, and their metabolites. And I'll go into more of that as we go. Let's see. So there's some references that'll be helpful for anybody that wants to dig a little deeper into this. So I have some of this work gets started with a paper I wrote back in 2014 on metabolic features of the cell danger response. We took up metabolomics and mass spectrometry and really to try to decipher the language of chemistry used by cells to coordinate their activities. And that's illustrated in part by a paper on myalgic encephalomyelitis and chronic fatigue syndrome. And then we have papers on the healing cycle, on environmental pollution activating the cell danger response. And to dig deep into the healing response of recovery on mitochondrial regulation of salugenesis, there's a paper from 2023. I think, and I'll mention the word suramin because that'll be coming up. It's a non-selective purinergic antagonist that we use to deconvolute some of the signaling systems that we study. The concepts of the cell danger response and salugenesis were born out of the practice of mitochondrial medicine. That's an important thing to remember. And every discovery we've ever made in the lab actually was inspired by a patient who was struggling with a problem. And that gave laser focus to some of the challenges that we had to overcome. Okay, so mitochondria change in response to infection, in response to learning and healing. And let's see if I can make this. So down here is that mitochondria are naturally social organelles. They live in a dynamic network that is interconnected and constantly breathing and changing in response to environmental signals and coordinating the messages that they create and send back to the nucleus and then outside of the cell to coordinate intercellular functions. So this is a picture of mitochondrial network, all connected. It turns out that if we injure mitochondria, in this case, what we've done is treated these cells with 25 millimolar glucose, Okay? And it leads to mitochondrial fragmentation. It also leads to the release of mitochondrial DNA with actually leaving an intact cell. This cell is not dead, but it's releasing mitochondrial DNA. And you may remember one of our earlier speakers talked about cytoplasmic mitochondrial DNA also being a danger signal. And that's definitely occurring inside here, as that mitochondrial DNA is released there as well. Well, so it turns out, okay, so this is an important concept. If mitochondria are blocked from adapting to stress, the cell dies. Mitochondria must change their activity over the course of the response to exercise, the response to nutrition, the response to an infection, in order to overcome and be able to retain metabolic memories that are hormetic and make those cells better able to handle those stresses in the future. So what happens when this is blocked? If we block the return pathway toward reestablishment of the mitochondrial network, then what happens is it leads to chronic illness. So it turns out that any symptom that lasts for at least 3 to 6 months, turns out that it's actively maintained by changes in mitochondrial function. And that'll be a topic that we will go into more. Okay? And when that happens, it leads to, so 60% of all humans on Earth under 65 have at least one chronic illness that they struggle with, and over 90% of adults over the age of 65 are struggling with at least one chronic illness. And now the statistics for children are about 40 to 50% of children struggle with a chronic illness. Biomedical research has progressed and has made great strides over the last 50 years by training investigators to focus on a single problem and to build a deep silo that defines how that disorder is different from every other chronic disorder. Okay? And I'm here to tell you it's the things that make us the same are more important than the things that make us different. And that is also true for chronic illness. So we have actually studied each one of these different disorders and looked at, we've used mass spectrometry to look at metabolic changes in everything from primary mitochondrial disease, was where we first got started, to autism, chronic fatigue syndrome, Gulf War illness, PTSD, Lyme, many of the other things that Rob Lustig mentions in this very kind introduction. But what we ultimately wanted to understand is what keeps people from getting better? What is blocking people from recovery? And one of the outcomes of the research is once you've identified the pathophysiologic trigger and removed it, it's no guarantee that a person will recover. Because recovery is a highly energy intensive process that cannot be completed unless your mitochondria are working properly and adapting to the conditions that are present. Okay, so let's see. So I was looking for what are the hidden connections? What are the similarities between these different disorders? And ultimately the conclusion was all of these different chronic complex disorders share the inability to complete what we call the healing cycle. And I'll go through what that is. Okay. A lot of our work comes from chronic fatigue syndrome and autism. So pathogenesis asks the question, what is the cause of disease? Salugenesis asks the question, What's the cause of healing? So we can look at diseases of acute injury and talk about what I refer to as the first book of medicine, which is really the corpus of medical knowledge accumulated over the last 5,000 years. And that's allowed healthcare workers and physicians to address a problem that can lead to rapid disability or loss of life. Okay, these are the things that can kill you fast, and they can be infections, they can be poisonings, they can be traumas of a variety of different kinds, but those are the things that medicine has spent its time really getting good at. What we're really bad at, okay, and so what this study of pathogenesis has allowed us to identify is that there are genes, there are pollutants, toxic metals, poor nutrition, microbiome, infectious and autoimmunity that are each outside influences that change the state of health to a state of injury. Okay? It turns out that in order to, and there are literally thousands of different ways that a person can become sick. And we can create long catalogs of all those things. But it turns out there's one conserved pathway that is agnostic as to how the injury occurred, okay, but utilizes the power of programmatic changes in mitochondrial metabolism in order to heal. And so this second limb is the process of recovery. And I want to emphasize this also difference in state versus process. So there is a choreographed sequence of metabolic events that must happen in order to, to begin the healing cycle. And it starts actually with something we call CDR1, cell danger response first phase, which uses M1 polarized pro-inflammatory mitochondria. This happens in all cells, healthy cells as well. If you cannot reprogram your mitochondria to become pro-inflammatory when they encounter a virus or significant bacterial infection, that organism will die. You need a pro-inflammatory response to contain the infection. Okay? That's why we talk about the yin and yang of oxidative stress versus oxidative shielding. So in these circumstances, the very first step of the healing cycle involves oxidative shielding and inflammation and a cascade of events that go from there. The second phase, CDR1, involves pluripotential mitochondria that allow cells to proliferate. Oh yeah, I'll go more into the different metabolic pathways later. But so this is a proliferative metabolism. Some people have learned the word Warburg metabolism. It's what is, it uses a pathway called aerobic glycolysis, but that's what's necessary to replace cells that were lost in the acute infection or the acute injury, okay? To recruit stem cells and to replace the biomass of cells that were lost. And then the next step is CDR3, which reestablishes the anti-inflammatory M2 phenotype of mitochondria in tissues. And that gets you back to what we call health star, which is a fragile state of recovered health. Let's see. Along the way, a person who is healing from let's say a concussion may go back to the game too soon and can receive a secondary head injury that even at a lower energy of impact produces a greater disability. Okay, so and exposure to the chemical exposome and all the endocrine disruptors and pesticides, obesogens that Dr. Lustig talked about also inhibit this process. But what's interesting about it is they not only cause an increase in reactive oxygen, but they cause an increase in the cellular release of ATP. Okay, and that ATP blocks further progress through this pathway. of salugenesis. Training and nutrition are the only ways proven to lead back to resilient health and increased fitness and longevity. In the brilliant talk that my dear friend Mark Tarnopolsky gave us just recently. So, okay. It turns out that incomplete healing after trauma, so if you block any one of these steps, you'll have cells that, remember, all tissues are mosaics of cells, okay? They're mosaics of interacting cell types. And if you block recovery, then it will lead to misrepaired injuries, cell loss, dedifferentiation, gliosis, then DNA repair abnormalities, all the different things that you associate with the aging process. Okay. All right. So what we're really interested in is discovering salugenesis therapies that unblock this progress through this universally conserved sequence of programmatic metabolic changes mediated by our mitochondria. Why don't we have any of these yet? Well, we just didn't know how to look. We spent really all of our time thinking about acute illnesses and not enough time thinking about what is different about chronic illness that makes it conceptually distinct from acute illness. Okay. So here are the metabolic features of the health cycle and the healing cycle. So ideally, we spend most of our time in the health cycle, which allows us to cycle through wakeful activity, nutrient intake, waste and toxin removal, social connection, nature connection, and restorative sleep. With general activity, there is a release of the right amount of extracellular ATP and reactive oxygen. Okay. And that ATP turns out to be metabolized down to adenosine, which is essential for initiating the sleep cycle. So the very thing that is actively produced by daily activity and nutrient intake is actually then metabolized as a signal that helps you initiate sleep. When we are exposed to stressors or pathogens or triggers of any different kind, it leads to 1,000 different possibilities for causing injury that I'll just call pathogenesis here. Okay. But a key feature of that is a dramatic increase in extracellular ATP release. So this is another thing that it's important to emphasize is that inside the cell, ATP contributes to all the housekeeping functions, cell growth, and repair that is needed throughout the day. But outside the cell, it has a completely different function that has nothing to do with energy. It signals danger. All right, so it turns out that if you stress a cell that has initially M2 filamentous, well-connected mitochondria, that within just a few minutes, let's see if this looks like we're not, okay, so I'll tell you what happens, is that these mitochondria begin to fission, okay? They will become, so here there's their filamentous anti-inflammatory M2 phenotype, but as soon as they begin to fission and take on a different structure, they'll become pro-inflammatory M1 mitochondria. Okay, so here are the phases. CDR1, actually, it's impossible for you to be inflamed without mitochondria being in CDR1. Macrophages that have a pro-inflammatory M1 phenotype have M1 mitochondria that are pro-inflammatory. Okay? This is a stage where mitochondrial oxidative phosphorylation must be dialed down, and the cell must turn up glycolytic energy production. So this is the phase where the metabolic program is glycolysis. Now, after the danger has been contained, then the cell must activate the growth program of CDR1 that requires Warburg metabolism and aerobic glycolysis. In CDR3, there's a reestablishment of cell-autonomous oxidative phosphorylation and differentiation of the mitochondria. And then only when we return to the health cycle do those cells become maximally entrained by brain safety signals so that the normal phasic rise and fall of metabolic activity occurs during the day and during the night, and then changes with the seasons. AMP kinase to mTOR changes in winter to summer metabolism, for example. Okay, so there are off ramps in this pathway, and one of them we call the hypometabolic survival state, HSS loop, and we believe that this is very similar to the C. elegans state that led to dozens of longevity genes being discovered. That state is called Dower. It is a state in which the worms now can quadruple their lifespan, okay, when they have been exposed to threat. And instead of living 1 month, they can live 4 months. Okay. And interestingly, the biological clock slows during that time. And only when the danger has passed and they re-enter the normal growth cycle do they re-enter their life cycle, and the chronologic and biological clocks begin to synchronize again. Okay. All right. So there are connections between the different phases. Post-exertional malaise, or PEM, is something that's common with stress in a patient with chronic fatigue syndrome. But this whole process together we call salugenesis. Is the return, and the goal of the healing cycle is return you to the health cycle. But if you get stuck in any one of these stages of the cell danger response, it leads to chronic symptoms of chronic illnesses. All right. So just to reiterate, what is the cell danger response? It's governed by over 5,000 genes that have been characterized by studies of patients who have been in sepsis, have suffered burn injuries or traumas of a variety of different kinds. Over 5,000 genes are known to be part of this, and over 40 key metabolic pathways are involved. This is not a one-trick pony phenomenon. The CDR is a universal response to environmental threat that's triggered by extracellular ATP. And the thing to remember is that healing can't occur without it. You absolutely need the cell danger response. You just don't want it to persist. It begins with mitochondria in the cell, but spreads in a signaling cascade to neighboring cells and up through afferent signals back to the brain through the vagus. So many of you may know already that the vagus nerve contains, 80% of the fibers in the vagus nerve are sensory fibers sending sensory information from the body to the brain. Okay? Only 20% are motor fibers that are moving from the brain to the tissues. Okay? It's biologically expensive. It is highly energy and resource intensive. And until the CDR is resolved, athletes and soldiers will underperform, aging is accelerated, and that symptoms of chronic disease and disability will persist. So the acute CDR is normal and necessary, but the chronic cell danger response that persists is abnormal and produces the symptoms of chronic illness. Okay. So again, what is the archetypal signal that recognized by literally all life on the planet, both prokaryotic and eukaryotic? It turns out that it's extracellular ATP. So here's an example from the humble mustard plant, Arabidopsis, that's been engineered to fluoresce in response to the release of activating amounts of calcium in the cell. And down, let's see if I can do this. So right here is a little cricket bite. And if I add ATP or glutamate, turns out both of these will do it. If I add ATP right here, then this is what happens. Ooh, an animation worked. Okay, yeah, just let's see, just to do that again. Okay, so within 90 seconds, that injury is communicated to the other side of the plant. Okay. Now, what happens if you put ATP in a little drop of a plate of microglia? These are inflammatory cells in the brain, and this is what happens. The microglia recognize this as a danger signal, and they send their pseudopodia to contain the threat. Okay. This can be inhibited by purinergic antagonists like suramin. Okay. Jeff Bernstock deserves the credit for having discovered this entirely new field of signaling. He's was one of the most published neuropharmacologists in the world. And back in 1972, he was using an intestinal smooth muscle assay and measuring the strength of the muscle contraction when exposed to a variety of different neurotransmitters, including ATP. And what he found is that he found that initially, it turns out there's a small blip of contraction followed by 30 seconds of relaxation followed by 3 minutes of contraction. Okay? And this was not blockable by atropine, which would have blocked any acetylcholine receptor signaling, and not blockable by guanethidine, which would have blocked norepinephrine signaling. All right, so ATP is a key regulator of the cell danger response. This is just a cartoon of mitochondria in the cell doing their thing. Mitochondria, oh yeah, so this is another important thing. So the mitochondrial proteome has about 1,134 proteins. 1,000 of those are made by genes in the nucleus. Well, actually, all but 13 are made by genes in the nucleus, and mitochondrial DNA only encodes 13 proteins. So the mitochondria are always tailored to the gene expression needs of the cell type in which they reside. There is a different specialized mitochondrion for every cell type in the body. So this is just an example of, so these cells under stress will release ATP and other metabolites. The cells will release exerkines. They will release mitokines and metabolites through channels that open up under redox stress. So pannexin and P2X7 are channels that will open up under changes in actually the lipid composition of the membrane in coronary arteries and endothelial cells. When there's hypertensive stress, the biomechanical stress will open these pathways. ATP is released from the cell, and oh yeah, and then in addition, to the human equivalent of EGL20 that we learned about as one of the mitokines, there's short path retrograde signals to the nucleus. The nucleus talks to mitochondria. There's a long path retrograde signal that moves from released metabolites to their receptors on the cell membrane. But when these channels open in the membrane, there's a dissipative loss of ATP. And that's where these two different avatars of ATP can be illustrated. So inside the cell, ATP is a metabolite, an energy carrier. Outside the cell, it is information. So there's a matter to information transformation that occurs when ATP is inside the cell to outside the cell. So we were looking for drugs that could block that loss of ATP and also block the binding of that danger signal to cellular receptors. There are 19 different purinergic receptors. Seromin binds about half of them. Okay, so just to remind you, ATP is this molecule. This is the purine moiety. This is the triphosphate. It's the basis of of the word purinergic signaling. It is the most expensive metabolite that cells make. Inside the cell, it's energy carrier. Outside the cell, it's information. And the receptors, P2Y, P2X, P1 adenosine receptors, are on every cell in different amounts and overlapping sets that are specific for the differentiated cell types. And I'll talk about that toward the end of this. Oh yeah, so I wanted to emphasize this importance. So a lot of people talk about, neurotransmission and neurotransmitters, but it turns out that ATP is co-released with every neurotransmitter that has been studied to date. So every time you look at a dopamine synapse or a serotonin synapse or a GABAergic synapse, ATP is also being released. All right, so when we did a lot of mass spectrometry and looked at a lot of different diseases, what we found is a core set of pathways that we use to ultimately that were the same in all these different diseases. And these pathways are listed off to the left. It's not one pathway. Okay? These are multiple things that turn out to each be affected and improved by the things that have already been talked about, like exercise, balanced nutrition, good sleep. All of these things, are dysregulated under these conditions of chronic illness. But I mean, it's even things as like obsessive compulsive disorder and traumatic brain injury, Lee syndrome, a classic mitochondrial disorder, all show these similar shared pathway disturbances. Okay, so it turns out that if you do a search for the genes that are related to ATP signaling through G-protein coupled receptors. So we have about, let's say, 800 or so G-protein coupled receptors that got their start actually as a family of genes in oceanic plankton that responded to light. These were the opsins. Okay? And they ramified, duplicated, and differentiated, and gave us what are called G-protein coupled receptors that regulate olfaction, that regulate pheromone signaling, that regulate taste, but also response to ATP signaling. And so they're, let's see if I, okay, so these are color-coded. These are, GPCRs that are, that responsive to Krebs cycle intermediates and nucleotides, signaling lipids, ketones, lactate, niacin, butyrate. Many of these you've already heard about. Turns out even viral co-receptors and anti-inflammatory peptides are also related by DNA sequence to purinergic receptors. And the angiotensin receptor 2 that is used as a viral entry site or viral entry receptor is related to also these P2Y1 related purinergic signaling GPCRs. And we know a lot about these. So we know what the canonical activation site looks like, and we know how certain drugs will inhibit the function of these GPCRs. And so in principle, would be possible to develop a shelf full of related drugs that would be selective for the 19 different purinergic receptors. A key concept that I want to keep hammering home is that stress starts with reactive oxygen and ATP release. That ATP release then binds to receptors on every single cell type in the body. And because of the differentiated functions of those cells and the different specific receptors, they each will release different mediators in response to the very same stimulus of ATP signaling. So in the case of mast cells, the ATP signaling causes release of histamine, tryptase, kinase, IL-6, and 100 other things, okay? Depending on the location of that mast cell, okay? Microglia, when they receive the signal from ATP, release IL-6 and IL-1 beta and MCP1 and reactive oxygen. Intracellular calcium release in cortical neurons. Also, it lowers the seizure threshold for and is involved in epilepsy. Acute inflammatory cells like neutrophils also have these receptors, and they will actually end up producing. So in this case, this line goes to here. It's chemotaxis and NADPH oxidase activation. Anyway, going all the way through T cells, Tregs, B cells, NK cells, macrophages. I want to get down to skeletal muscle. ATP signaling inhibits oxidative phosphorylation, okay, in a number of different ways. It also, when cells in the muscle release ATP, they recruit T cells and macrophages into the muscle. And so you'll see migrating cells coming into the muscle. All right. So the last few slides. In this set of slides, I'm trying to illustrate the point that adoptive transfer experiments where we take the plasma of a patient who has a chronic illness and treat cells in a dish that are otherwise healthy, we can recapitulate the mitochondrial phenotype of these illnesses. Some of the mitochondrial phenotypes. So Ron Davis developed a cellular impedance method called a nanoneedle and published in PNAS, where he showed, on the one hand, if you took peripheral blood mononuclear cells from a patient with chronic fatigue syndrome and put them in this chamber with a little bit higher salt to stress them, within 1 to 2 hours, they actually lost their impedance. Okay, lost the resistance across the membrane. He also and in addition to that, if you flip the experiment and you take healthy PBMCs and expose them to the plasma of a patient with ME/CFS, it produces the same phenotype, although slightly attenuated. So he also showed that red blood cells from patients with chronic fatigue syndrome have stiffer membranes and have a harder time making it through capillaries and a microfluidics chamber without some damage. And it turns out that if you flip that and take healthy red blood cells and expose them to the plasma of a patient with ME/CFS, you also stiffen the membrane. Bhupesh Prusti and I did an experiment where we took the plasma of patients with ME/CFS and transferred that plasma to cells in a dish and produced mitochondrial fragmentation, hypometabolism, and interestingly, an antiviral effect. The cells that had a decreased mitochondrial oxidative phosphorylation turned out to be acutely resistant to an RNA virus, influenza A, and to a DNA virus, herpes simplex virus 1. Okay, so, and we have a paper in peer review right now that's showing that certain immune complexes in the plasma of patients with ME/CFS produce the same mitochondrial hypometabolism and fragmentation. And in the last few slides, I'm going to show you that's done with Himal Patel from UC San Diego. We take plasma from patients and in chronic fatigue syndrome, actually leveraging our study from 2016 and showing that when we treat muscle cells in a dish, we can reproduce this mitochondrial hypometabolism. So to quantify that, we used a seahorse analyzer that measures oxygen consumption. We quantified basal respiration, ATP-linked respiration, as well as maximal uncoupled oxidative phosphorylation. And what we asked this question, are there blood signals that actually trigger fatigue via their effects on mitochondria and will produce a change in the normal filamentous network of mitochondria to fragmented organelles? Okay, so this is a study that looks at the oxygen consumption rate, and red is the chronic fatigue syndrome patients, blue are the controls, and in each case, males, females in total, there was a decrease in maximal mitochondrial respiration. But not only that, it turned out that it's not as simple as just turning down mitochondrial oxidative phosphorylation. Their capacity for glycolytic energy production was also diminished compared to controls. So this is also, the red is also decreased. Okay. So what's the scientific summary? The mitochondrial dysfunction that we observe in chronic disorders like chronic fatigue syndrome and autism is not actually a dysfunction. The mitochondria are doing precisely as they were instructed by signals in the blood and from the brain. These mitochondria, at least in the shorter term, are not damaged, diseased, or terminally exhausted. Their functions are being actively changed by signals in the blood, and this physiologic state can be traced back to ATP signaling. So there's hope, and mitochondria, and patients with chronic symptoms traceable to mitochondrial dysfunction can recover through a variety of different ways. But these results establish a mechanistic foundation and incentive for accelerated development of anticholinergic drug as a new class of medicines in our pharmacopoeia. The world's pharmacopoeia does not have medicines that you can take off the shelf and use to regulate this signaling pathway. Okay. And so it'll desensitize ATP hypersensitivity. I'll actually emphasize this point of hypersensitivity because adaptive changes are associated with a sensitization of cells to smaller and smaller amounts of stress, and as regulated by, or registered by smaller amounts of ATP release. So you need to desensitize the cells. And then ultimately, several other chronic disorders show similar biology, including long COVID, post-Lyme, post-chemo, autism, early stages of Parkinson and Alzheimer. So I'll end with a little plug here that I believe that the medical dark matter of the 21st century is salugenesis, and that abnormalities in the process of salugenesis affect mitochondria and are present in over 90% of chronic illness. So we spend all our time talking about pathophysiologic changes, but so far we've had blinders on this highly conserved process of recovery called salugenesis. And I'll leave it there. And thank everybody and the financial supporters, and particularly the UCSD Christini Fund, a little girl with Lee syndrome, who inspired all this work back starting in the 1990s. So that's it, and thank you. Questions?
Stay tuned for the second annual Symposium on Mitochondrial Health, Health Span, and Aging.